All right, everyone, let's talk today about the cells and the intrinsic conduction in the heart. Right? Let me start by going to my PowerPoint. We'll discuss some of this. It's going to be a bit of a review from skeletal muscle, but some of it's going to be new. It's a really fun topic. All right. So what's different between skeletal and cardiac? Here you see a picture. Can you see up toward the top here? You can see the skeletal muscle histology. You can reach back in your memory to, it says cardiac muscles histology. Can you reach back in your memory to the skeletal muscle? What does skeletal muscle look like? Well, it looks here like this cardiac myocyte starts here and ends here, right? Did the skeletal muscles do that? Or did they reach the whole length of the muscle? Right? They reached the whole, they traveled, they stretched the whole length of the muscle. Cardiac myocytes are short. And look at this. If you can watch the mouse, it starts here and it ends here and here. So they're branched. There's two different branches. They're short and they're branched. Now, what keeps them from pulling apart? Actually, maybe I should go with the PowerPoint. What's kind of missing? Doesn't the sarcoplasmic reticulum down here look thinner? Remember how extensive that was in skeletal muscle? Here in cardiac, it's a lot thinner. Isn't that surprising? Because my cardiac muscles are working so much harder than my skeletal right now. Well, in general. All right. And what else do we see? You see down here in the bottom picture, ions are coming through these little pores. Do you remember back from chapter 3, gap junctions? Isn't that interesting? We still have striations, but look at these thick lines right here where I was talking back up in the histology picture. See how these thick lines here, where it starts, where it ends, those are called intercalated discs. That's something you really want to know. Intercalated discs are the boundary between the two different cardiac cells. So here I have some things written down here, but let me do intercalated discs. And in those intercalated discs, that's where you're going to find gap junctions and desmosomes. Remember from chapter 3, we talked about both of those then. All right, so thicker sarcoplasmic reticulum. What did the sarcoplasmic reticulum do? Stores calcium ions, right? So cardiac muscle must have something else that it's going to use for calcium or some other way of getting calcium, right? All right, the cells are long and thin and skeletal, short and branched. And actually, they usually only have 1 or 2 nuclei, whereas in skeletal muscle, remember, they had a lot of nuclei. All right. Our voltage-gated channels are going to be somewhat similar. You're going to have sodium and potassium, just like you did with skeletal muscle. But, oh, look at this. We're also going to throw in calcium. So that makes sense, since the sarcoplasmic reticulum in cardiac is thinner. So interesting, right? But we're also going to throw in something else, HCN, these positive ion leak channels. We'll talk about that. Those are just in the pacemakers. Now, remember we did have leak channels that maintain resting membrane potential. Pacemaker cells are going to have something special with these positive ion channels. All right, I need to share my screen first. All right, let's go back to the PowerPoint. Okay. Oh, here we are. Oh, much better. All right. So we talked about what the sarcoplasmic reticulum is. Here you can really see those intercalated discs, right? Intercalated discs. All right, and here you can really see those gap junctions. Here, the desmosomes. What do desmosomes do? They hold it together really tightly. If you're a cardiac cell and you're contracting, you don't want to rip away from the cells next to you, right? So it's kind of like a tight hold there. Different from tight junctions are to prevent leakage more. Desmosomes are mechanical strength. Alrighty, get everything here. This is just comparing the three tissue types. Skeletal muscle, look at those nice, beautiful striations. Cardiac, you still have striations, but what you really notice here are these intercalated discs. Also, I want to point out, where are the nuclei in cardiac? Right in the middle, right? How about here? Right on the edges for skeletal muscle. Here's smooth muscle, no striations. All right. Something else that is really fun in cardiac tissue, it's myoglobin. What is myoglobin? What could it be? Let's pull it apart. What is myo? Muscle, right? Myocyte. What is globin? What does that sound like? Does that remind you of anything? Hemoglobin, perhaps? So this is going to be holding oxygen. Why would a cardiac myocyte want to have an extra supply of oxygen? Well, because it's contracting, and it's not like my heart muscle is going to stop and rest for any long period of time, right? So we need to have a good supply of oxygen. You need to keep it well supplied. Okay. Also, let's talk about the neuromuscular junction. Did we see one in those pictures? Where is that? And don't we need neurotransmitters to bind to these ligand-gated channels to initiate a contraction? We seem to be missing that, didn't we? So how do these cells know when to contract? Pacemakers. Pacemakers. They tell the muscle cells, the cardiac muscle cells, contract, contract, contract. Pacemakers contract without any neural input. It's really amazing. Let me talk to you about these. These pacemaker cells, they don't need input from the central nervous system to depolarize. In fact, if you took them out and put them in a petri dish with some culture media, which is like food and nutrients, they would just sit there contracting all on their own. But if they're not attached by gap junctions, they'll just be contracting and then not necessarily contract in rhythm with each other. It's those gap junctions that coordinate the contraction. Pretty interesting, isn't it? All right. But how do they do that? I mean, how does a cell just contract on its own, right? This is really fun. All right, so you have these HCN channels. Oh, we talked about those, didn't we? These special positive ion leak channels. And so they're leaking until, boom, they hit threshold. So these pacemaker cells leak. Positive ions leak in, and more leaking in than out here. And you reach, it slightly depolarizes, and you reach threshold, and then boom, that's when your calcium channels will open, not sodium, calcium. Interesting, right? So the positive ion leak channels depolarize, get you to threshold, and then threshold, boom, calcium channels open. Calcium floods in. And you see in the picture here, here's calcium opening, -40, and then that takes you up. the rest of your depolarization. And then just like in, well, similar to in skeletal, once you reach the peak, the potassium channels open and membrane potential comes back down. And as soon as you're back down, see how it starts over again? Potassium's going to close, and then cycle begins again with the leak channels. So leak, 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 boom, calcium opens, then sodium or potassium opens, and then after potassium's closing, cycle starts over. Let's see if I can say it. So we're going, there we go, leak, calcium, potassium, and then back to resting membrane potential. Leak, calcium, potassium, resting membrane potential. Leak, calcium, potassium, resting membrane potential. Leak, but leak's going to take longer. Leak, calcium, potassium, resting membrane potential. Leak, calcium, potassium, resting membrane potential. Leak, calcium, potassium, resting membrane potential. Does that make sense? And what cells are these? Are these your regular heart cells? No, these are your pacemakers, right? Pacemakers, leak, calcium, potassium, resting membrane potential. Leak. Okay, well, you get the picture. Remember, these are these are the pacemakers. They only account for about 1% of the cells in the heart. All right. So let's go back up here. What cell is actually doing the depolarization, or not what cell? What channel is doing accounts for the main depolarization? Calcium, right? Whereas in skeletal muscle, it was sodium. And also, what's getting us to threshold? It is those leak channels. What's the only thing that's truly similar here is that potassium's bringing us back to resting membrane potential. All right, that's the one thing that's the same here. All right, so after these pacemakers depolarize, what happens? Okay, so see here in this picture, you have your sinoatrial node, the SA node. Remember, these are the pacemakers here. So you're going to have these conducting fibers that are going to come down from the SA node, where it starts, down to this here, the AV node. Now take a look at this structure here. They also go to the other atrium, so that the atria can contract in unison. But notice how you have these conducting fibers going down to the AV node. Do you have a lot of them? In the picture, we have 3, right? How many fibers do we have coming out? Only 1. So if you have 3 lanes on the highway going into an area and only 1 lane coming out, what happens? Does traffic get faster or slower? It gets slower, right? So there's going to be this AV node delay. It doesn't necessarily cause the electrical impulse itself to be slow, but it makes it pause. We call it the AV node delay. So here's the atrioventricular node, and there's a delay here. All right. So you're going to go down the AV bundle, AV node, AV bundle, the right and left bundle branches, and the Purkinje fibers. All right, but I want you to know about that AV node delay, because that's going to be significant. Let's talk about it for a second here. All right, this AV node bundle, we have this, what is this electrical impulse going to cause? It's going to cause the contraction of the cells, right? Which is going to cause the contraction of the heart. So we want the atria to contract first, right? Get the blood into the ventricles. Do we want the ventricles to contract immediately? Not really. We want them to fill with blood first. So the ventricles are going to fill with blood while the atria are contracting. You need that AV node delay. Does that make sense? It's a fun concept. All right, this is from an old book, this picture. but I really liked how it spelled it out. The SA node, sinoatrial node, AV node, which is the atrioventricular node, the AV bundle, the right and left bundle branches, and you can call these Purkinje fibers. So I believe that's a question in your guided notes, and it's probably in your lab book somewhere as well. All right. So far, what have we talked about? Structure of the muscle. We've talked about how a pacemaker depolarized. We talked about the sinoatrial and the atrioventricular nodes. And we talked about the pathway that the electrical impulses take. So let's consider what happens when these impulses get to the cardiac myocytes. All right. This is a whole picture. I know it's hard to see there. So let's go with the little ones. All right. So remember, the signal comes in by how? By a motor neuron? No. By ions traveling through gap junctions? Yes. And what's the point of these gap junctions? You know what it's doing? It's coordinating the contraction. Because I don't want one part of my heart to contract and then another part later. No, I want it to contract as a unit. So you have gap junctions when you need coordinated contraction. We're talking about it in the GI system. The uterus has them as well, at the end of childbirth. All right, so you want your heart to contract as a unit. Okay. So what happens first? Oh, this looks familiar, right? We have those sodium channels. Sodium channels open. Oh, and then look. Okay, so we depolarize fast, right? Potassium channels open. Oh, looks like we're going to depolarize. Go back to resting membrane potential. Are we? What's going to happen next? What's this green channel sitting here? What's calcium going to do? It's not doing anything yet, right? What's it going to do? Oh, look at that. So potassium opens, and then calcium opens right away. And it says, no, we're not repolarizing yet. And then, so calcium stays open for a bit, about 200 milliseconds, and then it closes and more potassium channels open, and you can go all the way back to resting membrane potential. Something I want to point out. I know those of you who had me for 141, we talked about the wave for skeletal muscle contraction, right? Sodium, potassium, sodium, potassium. So here, it's sodium, and then, it's hard to do it on video. Sodium, potassium starts, but then calcium comes, it holds it. And then potassium can repolarize. So sodium, potassium, calcium, potassium, sodium, potassium, calcium, potassium. Does this make a little bit of sense? Let's go back to the PowerPoint here. All right. So what happens here is, see this refractory period? You have a much longer refractory period in, there we go, in cardiac cells than you do in skeletal muscle. So here we go, sodium, a little bit of potassium, calcium holding it in place, calcium, and oh, let's talk about this. All right, sodium ions in, potassium ions out. Which way does calcium have to go to counteract the potassium? Calcium has to come in. And interesting, right? So what's the point of calcium coming in? Well, we actually get calcium that way, and it will induce calcium release. It will help with the calcium release from the sarcoplasmic reticulum, because we're going to keep that membrane potential. All right. So let's talk about the difference here again between skeletal and muscle. All right, skeletal, you have sodium, potassium, right? Sodium, potassium. Here we have sodium, calcium. Oh, well, let me stop. Sodium, potassium, calcium, potassium. Sodium, potassium, calcium, potassium. Does that make sense? You get that long refractory period. Let me draw something on the board here. What about, remember we talked about tetanus back in chapter 10? When you get enough And you had this continual, you know what, I need a different color there. You had this continual contraction. Can you have that in cardiac? When cardiac's, no, you can't, right? It has this forced refractory period. Skeletal muscle, and here's cardiac muscle. Does that make sense? So you're not going to get tetanus. Why is that important? Well, we don't want to get tetanus in our cardiac muscle because we need it to pause. We need those ventricles to fill. It's just like the AV node delay plays a role in this too. But the cardiac myocytes is kind of built in with those calcium channels. Contract and then fully relax. Repolarize. All right. All right. And so why is this important? We kind of just discussed it, but we need the blood to get from the atrium to the ventricles. You need time for it to fill before it's going to contract again. So that's a lot of the point. We're going to discuss this more. I anticipate answering questions about these calcium channels. But thanks for watching. See you guys tonight.